Okay, let's get started. Hello, everyone. Thank you for joining the Marfan Foundation and its divisions for our webinar titled How to Read a Genetic Testing Report, a Practical Guide for Individuals and Families. I'm Lauren May, Senior VP of Research at the Foundation. I'm joined by my colleagues, Dr. Josephine Grima, who's our Chief Science Officer, and Stephanie Amdur-Clark, who's the Director of our Help and Resource Center. Before we begin the presentation, Helene Baruch, the Marfan Foundation's Deputy Executive Officer, would like to say a few words. Helene?
Thanks, Lauren. Welcome, everybody. I'm seeing we've got people from all over the country, which is very exciting. Welcome to our second webinar of our three-part series on genetics. We had one a couple weeks ago. Thrilled to have Dr. Byers join us tonight. And our third one is going to be, Lauren, I believe, March 21st, correct?
That's right.
So I hope that you can join us as well. Feel free to send notes in the webinar chat. We'll have some fun. We're going to put some stuff in information in there and resources. And I just want to say we've had about 253 people register tonight. I believe we have about 65 on and growing. So thank you. We've got, wow, Ghana on. Look at that. If I'm saying that right. We've got North Carolina, Canada, Colorado, and Virginia. It keeps coming. This is great, guys. And don't forget, Lauren's going to talk a little bit about if you have any questions to put it into the Q&A, not the chat. And I'm just going to pass it back over and enjoy the session. And we're here for you.
Thanks, Helene. So as Helene mentioned, we will be using the Q&A feature in Zoom to take your questions. You can submit them anytime during the webinar by clicking the Q&A tab, and we'll answer as many as we can at the end. Also, this webinar is being recorded and will be posted to the Marfan Foundation's YouTube channel in the next few days. I am very excited to introduce our speaker for today. Dr. Peter Byers received a BA in anthropology from Reed College in Portland, Oregon, and an MD from Case Western Reserve University in Cleveland, Ohio. He completed 2 years of training in internal medicine at UCSF, 3 years at the NIH as a research associate working on collagen proteins, and 3 years training in medical genetics and biochemistry at the University of Washington, or UW. Dr. Byers has been on the faculty at UW since 1977, with a joint appointment in pathology, which is now called lab medicine and pathology, and medicine and medical genetics. He is also a member of the Foundation's Professional Advisory Board and Scientific Advisory Board. Dr. Byers, this webinar was actually your idea. You identified this topic as one that would be of real benefit to our community, so I thank you for bringing it forward and, of course, for being with us today. The floor is yours.
Thank you very much, Lauren. I have a cold, and so I don't sound quite the same as I used to. But I'm hoping that will not interfere too much with what we're doing. So this got started in part because Lauren and I were talking at one point about, and also with Stephanie, about the idea that reading genetic reports seem to be sort of a mystery to people. And it really, I don't think it is a mystery, but I've been doing it for a long time, so I'm used to it. So I thought that it would be helpful to try and introduce you to the vocabulary and the structure of reports. What's there? What should be there? When you see the report, what do you want to have in it? And then how to understand what it says. And reports are different from different laboratories. I'll show you some from ours, and I'll show you a couple from others. And you'll see that they're structured a little bit differently, but they have basically the same information, and they try to get you an answer. So be able to make it go forward. I'm stuck.
Will the arrow key work on your keyboard?
Yeah, the down, there it goes. Okay, great. So I have no conflicts. I've always wanted to be able to make a lot of money from things like this, but haven't. I've created this the Collagen Diagnostic Laboratory in the early 1980s, and I'm now the director emeritus of it. And it's an academic diagnostic laboratory, and that's really the only conflict that I have. So I like to start when I talk about genetics and testing and things like that with this slide, which is this is a representation of a very small part of the genome, the letters in the background, which I think is really the greatest book ever written. And you can always get a bunch of pushback about that. But it's a remarkable book in the sense that it's written with 4 letters, A, C, G, and T. And it really contains all of human history with the history of wars, plagues, pestilence, famine, and everything else included in that background with lots and lots of things having happened. And the thing that's astonishing about it is that in some instances, it's sensitive to a change of a single nucleotide. That is one building block. Each of these letters is a nucleotide. And in others, and that can give you a lethal form of osteogenesis imperfecta. And in others, you can lose almost a whole chromosome, or essentially a whole chromosome, and have Turner syndrome, which is a relatively mild condition. And how that happens has been of interest to us as geneticists and to you as people who take care of or are in families with genetic conditions. And I'm going to try to get to that. And I think that, there are books that are written that have, out of our 26-letter alphabet, use 25 of the letters and are written without an E. And that's thought to be a really an incredible task. This is one of them. And you may not notice that the letters, it's not using words with E's in them. But I think that to write a book that's as complex as what we have as a genome with only 4 letters is really a magnum opus. So this is an image of a genetic report. And I'm going to start you in the fire so that we can really describe what's here and what are the things that are important to you and to us when we put these together. This is from us as a Collagen Diagnostic Laboratory. And there are certain things that need to be in the report and the structure of how it works here. So the first is an identifier. And up here in the upper right-hand corner of the left page here is the laboratory identification number and the patient name, which I've redacted, director and so forth, and then the genetic counselor who's been involved in it. It's the final result, which is indicated here, and the molecular test result. And it's the result of genomic sequencing and deletion duplication analysis of genes associated with arterial aneurysms and dissections. So the sample type that was provided was blood. The referral diagnosis was a thoracic aortic dissection. And the indications for testing was an acute type B aortic dissection with contained intrathoracic rupture, ongoing bleeding from all sources during pregnancy. And the molecular test result is right here. That is a word description of it, which is a COL3A1 pathogenic variant identified. So the term pathogenic variant is relatively new to the nomenclature of genetics, probably appeared in the last 10 to 15 years. And before that, these were called mutations, and mutations were basically deleterious. So that's one of the things that you have to remember about this. So we sequenced a bunch of genes, and the genes are listed here. The gene names are all letters and numbers. And in human genes, they're all capitalized and they're written in italics. The encoded protein is written here. So ACTA2 gene encodes the ACTA2 smooth muscle cell alpha actin. BGN encodes biglycan. And COL3A1, which was the gene here, encodes the pro-alpha 1,3 chains of type 3 procollagen, in which a pathogenic variant was identified. And for all the other genes here and then over here, there was nothing identified in them. And no abnormalities were identified. So the meat of the report is in this box, or a series of boxes here. And it says, first of all, heterozygous pathogenic variant. So heterozygous, we have 2 copies of every gene. And so they're different in the sense that one copy of the gene has an alteration, and the other copy of the gene is wild type or is normal. So the gene is indicated here, COL3A1, and it encodes, as I said here, the chains of type 3 procollagen. There are a number of different iterations of the sequence of the coding sequence of the gene, and they are contained and they are identified by this NM underscore 0000990.4. So 90 is always the COL3A1 gene, and 0.4 is the edition in which that occurs. The mutation occurs in exon 20, and you see there's an asterisk there, and it says this is exon 19 in the reference sequence. We've used a fibrillar collagen-specific legacy nomenclature that's designed to unify the description of variants in the triple helical domain and have a standard for the naming of exons. So that means when we compare different genes that are collagen genes, type 1 collagen genes, and so forth, that we want them to have the same exon numbering. So this is the DNA change. It's actually the coding sequence change. C.1304G, and this looks like greater than A, but it's actually just a short arrow, and it means a G's changed to an A in one copy of the gene. And then P means protein sequence, period, dot. Gly in the 3-letter code, glycine, at 435, substituted by aspartic acid. And if the triple helix is a region of the collagen molecule which extends in this gene from position 168 out about 1,029 amino acids. And in that position, it's the glycine 268 in the triple helical domain. We use that so that we can compare across other genes. The type of alteration is a substitution. That is, there's a substitution of an A for a G in one copy of the gene, and that's called a missense. So missense means it changes the sense of the amino acid from a glycine to an aspartic acid. That is what it encodes. A nonsense mutation is one that changes the wild type amino acid to a stop codon. That is, it says stop making protein here. Then there's the interpretation, and it says our findings are compatible with the clinical diagnosis of vascular Ehlers-Danlos syndrome. The studies identified a pathogenic variant, see the table, in one allele, that is one copy of the COL3A1, the gene that encodes the pro-alpha 1,3 chains of type 3 procollagen. And then it gives a series of much more information here, more information about this. Now, there's a third page that I didn't include that contains more information. It gives information about how the sequencing was done, what kind of things are important. And what you see is that we have here our address and our contact information. And then on every page, there's the identifier of who it's about, the date of birth. So every sample that comes in needs to have 2 identifiers, a name usually and a date of birth. And so we make sure that it's the same. And the pages are numbered, and this is page 2 of 4. And most of the reports do that. They don't always have it as page 2 of 4 in the sequence, but they have a numerical number. So there are a number of things here that can be confusing. The coding this, where YC, YP here, and then Y 3 letters here, and so forth. So the next slide. So let's go back one. And so you'll see something that's unusual, that there can only be 4 letters in this box, G, A, C, G, and T, but there are 20 different possible amino acids here. And it can be confusing because in some reports, the single letter code for amino acids are used, and there's an overlap. So the 4 nucleotides, A is adenosine, C is cytosine, G is guanine, and T is thymine. And these are the only building blocks of our genome, only building blocks. There are 20 amino acids, and I will show you how these are related to each other. And there is a single letter code for the amino acids. A, in amino acid terms, is alanine. C, in amino acid terms, is cysteine. G, in amino acids, is glycine. And T is threonine. So they have very different meanings when they're used. And so we choose to use the 3-letter code, which is here. ALA, ALA is alanine, cis is cysteine, asp is aspartic acid, GLU is glutamic acid, phe is phenylalanine. Phenylalanine, I left out an L. Gly is glycine, H is histidine, ILE is isoleucine, K is lysine, L is leucine, which is related to isoleucine, but different. M is methionine, N is asparagine, instead of aspartic acid. O is not used. Pro is proline, glutamine, arginine. Arginine is R, arginine. Serine, S. Threonine, and then valine, tryptophan, and tyrosine. So Z, X, U, O, J, and B are not used. O is occasionally used in some papers as a hydroxylated form of proline, which is common in in the collagen molecules, but there's not a codon for hydroxyproline. The hydroxylation is post-translation. So you have to be careful in how you use and make sure that you know. Now there's a different format. So if you notice in the C or coding sequence or DNA change, the position in which it's altered comes first and the G to A comes afterwards. In the protein position, the normal sequence, the wild type amino acid is first, then the position, and then the substituting amino acid. So they have a different format, and that's important to learn. And then here, this greater than sign or arrow means that G is substituted by A. And over here, the fact that there's something after the number means that the aspartic acid is substituted for glycine. So one of the things to notice is that in order to really identify where things are, you have to have a fair amount of information. So this is the coding sequence change in something. Okay. C 20 A to T, C 3190 G to T, then C 718 G to C. Well, if you ask me my address and where do I live, if I tell you 1523 and stop there, you don't have any idea where I live. You don't know what city I live in. You don't know what street I live on. You don't know what state it's in or anything. So if you give me this information in a report or a letter or a request, I don't know what you're talking about. And I have to infer what the gene is and all that sort of stuff. So this number helps because this identifier is unique. And I can go to a table and I can look up and see what it is. It's a little bit easier, or it helps a little bit more if I see what the substitution does. So in this case, at position 20, of the coding sequence. It's amino acid 6, and it's a glutamic acid to valine substitution. This is a very famous mutation. It was one of the first mutations ever identified in human DNA. This is a glycine at position 1064 to cysteine, and this is an arginine 239 to proline substitution. But we still don't know. We don't have the gene name yet. Now we have the gene name. This is hemoglobin B. This is type 1. This is the COL1A1 gene of type 1 collagen, and this is fibrillin, FBN1. And they each produce a different phenotype. So this was the first, this is the gene, the genetic change in beta globin that causes sickle cell disease. This causes a form of OI that was the first single nucleotide substitution in collagen that was identified. And this produces a Marfan syndrome. And I think this was the first Marfan mutation identified. So you can see how you get, how getting all the information gives you much more sense of what's going on. So why do people have genetic testing, and what's the consequence? So sometimes it's a family member has a known genetic condition, and you can test usually with a single nucleotide testing, although that's not usually done. You're thought to have an identifiable genetic condition, but say it's an aortic disease. So single gene testing or panel testing using genes that encode similar conditions can be used. You're thought to have a genetic cause for what you have, but no specific diagnosis. And in that case, a whole exome or genome sequence. So the exome is all of the coding region of the genome, and the genome sequence is everything else as well. And sometimes preconception genetic screening for recessive conditions, where panel testing is done for several hundred genes, and then or concurrently testing with a partner is done as well to see if there's a risk for having a genetic cause. So many of the genes that we deal with about aortic aneurysms are inherited as having one alteration, one copy of the gene that's altered, like COL3A1. And when you have children, you can either pass on that gene or pass on the normal ones. And so there's a 50% risk of having an affected person. But if you're this person who's apparently unaffected, you don't know. And so you might want to be tested. Or you can do prenatal testing in this case, where we could show that this kid was unaffected, pregnancy was unaffected. So other conditions are recessively inherited, like cystic fibrosis or sickle cell disease. And in that case, the affected people have two altered copies. They may be exactly the same, which is true for sickle cell disease, or they may be different, which is often the case with cystic fibrosis. And if you want to know, so is this person a carrier? Because it is presumably in this context it's not affected, but did this person receive the altered copy of the allele from this parent? And if so, then is this person also a carrier? And then what can they do about testing? So the questions are different, but the genetic alterations are the same. So one of the things that I think is very confusing to people is how are genetic alterations described? And there are basically three outcomes. First, it's causative or pathogenic. The second is it's not causative, or in this case, benign, or we don't know. And we don't know for a variety of reasons. Sometimes it's because we've not seen it before, and so we don't have enough evidence to know. There are alterations that are probably pathogenic, but we just don't have quite enough evidence to say that. And the others that are probably not causative or probably benign don't have quite enough to say. But the confusing part really is in these ones that are of uncertain significance. That is, we really don't know whether the alterations we've seen are genetic. So what are we dealing with when we're doing this? So the genome is about 2 meters of DNA, all in a cell that's the size or smaller than the tip of a pencil point. And so it gets wrapped up in a very complex fashion. And the DNA is spread among 23 pairs of chromosomes in women, or 22 pairs of pairs in men, women have two X's and men have an X and a Y. And these are chromosomes that have been isolated and kind of stretched out. You can see there are two copies of every chromosome along the way, and you can tell that this is from a woman or a female because it's XX. Here is an assessment of how much DNA there is. So our total genome, which is down here, is 3 billion nucleotides times 2, because there are two copies of it. And the chromosomes differ in size from 250,000 or so, or 250 million, to the Y chromosome, which is a squidgety little thing of only 57 million. The numbers of genes, protein coding genes that are on there differ from a little over 2,000 to only a few on the Y chromosome of 71. There's a lot of other stuff, pseudogenes, that is genetic material that looks exactly like or very similar to the coding genes. And you can see that almost 2/3 of the sequences of the real coding genes are represented in pseudogenes. And then there are a whole bunch of other things. So all of this other stuff used to be called junk DNA, but it's now really the regulatory sequences that are there. So if you look at what these sequences are like in a gene, there's a bunch of stuff up here that controls when and in what cells they're going to be. This gene is going to be made. So those 20,000 genes are not all encoded or not all expressed in every cell in the body, but only in some. They're structured in such a way that there's a bunch of stuff here that determines where all of this is going to happen. And then the coding sequence for the protein starts here and is encoded in the red part of the gene. And then there's a little bit of stuff that comes afterwards. The coding sequence is interrupted. So these red things are called exons, and the gray things are introns. But in order to make a protein and to read it so that you get everything right, all the stuff in here has to be clipped out. And so you have the protein coding region. And that's then translated by a ribosome to make a protein, which is a completely different structure. This is all made of amino acids. All of this stuff up here is made of nucleotides. So there are about 180,000 exons in the 20,000 genes. And that is contained in about 5% of the entire genome. So we're only looking at 1/20th of all the genetic sequence when we do exome sequencing. So this is a representation in letters that you can see. In the lowercase are the introns. The red and uppercase are the coding sequence or exon. There's information at the end of this intron and the beginning of this intron that basically identify the region that's the coding sequence. And then we get a protein that's here, and you can see it's a part of a collagen sequence with glycine, glutamic acid, gly-glu, ala, glyproglin, glypro-arch, glycir-glu. So it's got a repeating sequence that looks like that. So in order to make 20 amino acids, our genome, our cells, take the 4 coding nucleotides and use them in groups of 3. And so with that, you can get, if you count all of this up, you realize that you have 64 possibilities. And that means that the genetic code is actually redundant. So there are 4 glycine codons. There are 6 arginine codons. There are 6 serine codons that can be made in different ways. There are 3 stop coding codons, which tell you when to stop. There's a single methionine, and methionine is always the first amino acid of a protein when it's being made. And you can see that if you change the first nucleotide of a glycine codon, the codon is 3 nucleotides. GGT can become AGT or serine. It become CGT or arginine, or it become TGT and cysteine. And so you can see that with single nucleotide changes, which are the most common kind of thing that happen, you can get that. If you change the second position, G, to A, you get aspartic acid. To C, you get alanine. And to T, you can get valine. So this kind of change is all is what we see most commonly. You can get these other amino acids, but you have to change 2 nucleotides to do it. And these amino acids are quite different in shape, and so here are pictures of the shapes of them. The basic residues, the acidic residues, the other ones that are different, and some that are very small. And glycine is the very smallest residue here. So the sequencing is now done by breaking up the genome into short fragments and then capturing them, and then sequencing these very small pieces, and then aligning these pieces. It's basically like trying to find, names in a telephone book. So you align this piece, which is about 125 nucleotides, with the standard genome, and then you compare all of the things that line in this, and you can see that you have 2 different things that occur. You have the gray ones, which are the standard sequence, which are indicated by blue up here, which is a C. And then you have the red ones, which are the substitutions, which is a T at that position. Well, it turns out that this is one of the ones that doesn't change an amino acid in the PLOD1 or gene for kyphoscoliotic Ehlers-Danlos syndrome. And so, but this is how sequences are looked at and what they are done. This is called next generation sequencing. And you can see that you can get hundreds of these at the same position to get a very high confidence level. So you can get single nucleotide substitutions. They can be missense, so that you change a glycine codon, GGT, to an arginine codon, which is arginine, or to a termination codon, which is a nonsense mutation. You can alter the splicing so that you don't necessarily get the pieces of DNA that have the coding sequence in there correctly. Or you can create a frame shift. That is, if you delete this G, instead of going GGT, CCT, CGT, you now go GTC, CTC. And so you have a completely different amino acid sequence. And by chance, you will encounter a stop codon downstream within 20 codons. And so that's another outcome. So as I said, they're described as being pathogenic or probably pathogenic, or benign or probably benign, or of uncertain significance. And what goes into deciding that? Well, the first question is this alteration in databases? Have we seen it before? What about in the population? So if we look at a population of about 800,000 people who have been sequenced, is it there? If it's more common than the condition, then it won't be the cause. And that's one of the ways that we can decide about this. Or if it's half as common, it won't cause, because that would mean that half of the people who have that would have this thing. So it's only in the case where everybody has the same mutation that you can make that decision about that. So are there studies that show that this mutation affects the structure of the function of the encoded protein? So we were lucky in the sense that we started doing protein studies before we did DNA studies. And so we had a lot of information about what kinds of changes actually made a difference. And that was very helpful. The computational data, which is in silico, is basically the AI of analysis, which allows you to make models and to do things. And has become very helpful in doing things. The next thing is, does the variant appear only in the affected family members and not in the unaffected family members? Or is it new and not in the family? So if it's new, then it's likely to be something that's causative. And then we ask about what it does in the RNA and the protein to try and figure out where it fits. So if we go back now, and we can see here, you can see this is from Invitae. So here's the lab. It's now LabCorp Invitae. It started out as Invitae. It's a woman. We don't have we've blacked out the identification information and blacked out the number. Lauren Puryear is a member of our genetic counseling group. And there was a request to look at a familial variant, SMAD3, with this particular alteration, C, 230 C to T, and the protein being arginine 74 to tryptophan that is known to be pathogenic. So that's been seen in other family members. And here the result is positive. And I'm not sure, I don't like the positive negative result, but we don't ever use that terminology. We say that the familial variant was identified because they're both positive results, assuming that you did it right. One is, and positive always has this, wonderful feeling of being beneficial, and this may be not so beneficial. And so the same sequence is represented here. In this case, this is a different one. Same gene, but a different substitution. And here, the result was not seen. So the result is negative. And you have no result represented here. But the thing that can be confusing about this is that you read it and you say, well, it looks like there's a result that's here, and that's not it, and it's pathogenic. It does say not identified, not detected. But if you're not careful in reading this, you may miss all of that. So be careful when you look at these results and read them. So we come back to here, and this is a much more complex one where we actually had done additional sequences. But you can see here it's the SMAD3 gene. The mutation was in intron 2, C409 minus 9C to G. What's this minus 9 business doing in there? Well, the numbered sequences of the nucleotides are continuous across all of the coding sequence and do not include the numbering system in the introns. So what this means is that 401 is the beginning number of an exon, and this is at minus 9 or 9 nucleotides upstream. So it's up 9. And does it do anything? Well, in fact, we were able to do a test and say, yes, it does. It's predicted to result in exon skipping. So it says it now doesn't recognize this and does something different. So that's a different result, and it's got a different nomenclature here. And we would then say, well, what's the effect on the protein? And the effect on the protein is here because it's lacking 44 amino acids encoded in the downstream exon. So we have a number of ways of looking at things and a number of resources that we have. This is the UCSC Genome Browser. This is a diagram of the gene with the exons being the vertical lines here. This is the LOVD single gene database. This is a sequence of COL3A1. It's flipped from left to right. You'll see that the sequence runs this way and it's backwards. And I don't know how I did that, but I recognize that I did it. And then here's the intron from a region there. So that's from the LOVD databases, which has a lot of pathogenic variants. ClinVar is a database to which many of the large laboratories report all of their exons. It has a paucity of clinical information. And NOMAD is a big database of about 800,000 people who are ostensibly normal but are without genetic conditions that's available for comparing and trying to find whether the variant is present there. So that's me. You can reach me at this email address, edu. I've had a couple of things that are missing there. So, and now I'm happy to stop and answer questions. So that's trying to give you the vocabulary that we use, where it comes from, what it's like, and how to use it, and why things may be different from you and from other people.
Dr. Byers, thank you so much for your presentation today. I know this is a topic of great interest to our communities. Members of our audience have submitted several questions for you, and we will go through those now.
Great.
Okay. So this person's asking, when should additional evaluation beyond standard COL3A1 genetic sequencing be considered to assess for a null variant? Specifically, under what circumstances is analysis of type 3 collagen through skin biopsy and fibroblast culture appropriate after genetic testing? This person says that many of their friends have had experiences with clinicians that diagnose VEDS based solely on identifying a COL3A1 mutation without discussing the specific variant type and its functional impact.
Well, you've hit one of the points that I find very interesting in the world, and it's not limited to people talking about COL3A1 variants. So we first started using protein analysis, but it's an art form, and it's not it's an analog kind of testing in the sense that it requires a lot of interpretation, and it requires very experienced people doing it. And as a consequence of that, we stopped doing it because it's labor intensive, it's very expensive, and it's not as definitive as the genetic testing. So you can get null mutations in COL3A1 in a variety of different ways. You can delete the entire gene, and there are examples of deletion of COL3A1 with portions or all of COL5A2, which are next to it in both. And the deletion of COL5A2 doesn't seem to make much difference. You can get changes in single codons that change a coding codon from as a nonsense mutation that will stop the translation. Now, you'd think that would make a short protein, but instead it destabilizes the RNA, so the RNA becomes very unstable. You can get the same effect with mutations that occur at splicing sites. So, for example, the sequence upstream from an exon is usually T or C, then AG, and then you start the coding sequence and in collagens, in almost all the all the exons, the first nucleotide of the exon is a G. So if you change in the AG just before the exon, if you change that G to an A, now you have an AG sequence, and now that changes the splice site. It moves the splice site one nucleotide into the exon, and you get a frame shift, and the frame shift will lead to a stop codon. And so that's another way of producing it. There are also nulls that occur because they're protein nulls, and that is the protein doesn't get incorporated into the molecule, and those occur if they're beyond the triple helical domain of the type 3 collagen molecule, and they interfere with chain-chain association. So 3 chains have to get together, and there are sequences there that determine that. And if they're changed or altered, then you can get protein alterations. So a genetic test that says this is a stop codon or creates a stop codon, I think is a definitive test. And it says that's enough for saying this is a null mutation. And you don't have to worry about it. If the question is, I think I have a COL3A1 mutation because I have some of the features of vascular EDS, but nobody's been able to find something. One of the questions would be, should you do something more? And in the past, we might have done protein testing, but that's not as sensitive. And the real question would be, can we find something that occurs in the introns that either add sequences that destabilize the RNA or deletes sequences or does something like that that we haven't detected by the usual kinds of genetic testing? And the answer is, if the clinical condition is striking enough to warrant it, then we will go in that direction. So I hope that helps answer the question.
It does. Thank you. That's great. We have a question from someone asking specifically about a past UW report, but I could see this
Mm-hmm.
being a question for anyone who had genetic
Mm-hmm.
testing a number of years ago. So in this case, if someone has a UW report from 2011 that doesn't include the extended explanation that the sample report you showed today does, is it possible to request a new report that includes that level of detail? Like, how does that work if over time, having had genetic testing a while ago and the report isn't as
Right.
detailed?
If the report isn't as detailed, but is basically sort of looks like a letter, we can always write a new report that has it in the current formatting. Uh, and if a mutation was identified, and a pathogenic variant was identified, that won't change. But we can make it so that it looks like a more standard report.
Okay.
If
And
you're
someone
asking
does
about
their, okay.
if you're asking whether we might miss things, the answer is, well, yeah, we could have, maybe. Genetic testing is, but if we found something, then it won't change the nature of the report. If we didn't find something, we now test for more genes than we used to, and all of those kinds of things. So it would be possible to extend it to that.
Okay. And someone would reach out to their geneticist if they wanted to get the
Yeah, or
2026 report.
right, or they can go back to the report, which will have the contact information for the genetic counselors in the CDL, and talk to them. So Drew Leistert is now the genetic counselor.
Great.
And you
Okay,
would
great.
have dealt with Melanie Pepin perhaps in 2011.
This person in 2017 had a COL3A1 gene test. VEDS was suspected at the time because of specific symptoms. The missense mutation that was found was classified as a VUS, a variant of uncertain significance, and remains so until this day. Is there any chance that the mutation will ever be classified? What do you do other than wait?
So, um, hmm. so things are classified as VUS for a number of reasons, and when we first started doing these kinds of things, when we would see things, we called every substitution for a glycine in the triple helical domain pathogenic. And that was because we knew that at the protein level that those substitutions altered the folding of the protein and made a difference. It didn't matter to us whether we hadn't seen it before, and this was before the compilation by the ACMG, American College of Medical Genetics, people putting together the guidelines for variant interpretation. And so we would often talk with people who had had that sort of thing happen. So some variants are alterations and give rise to phenotypes which are similar to vascular EDS because they change. The reason is not entirely clear, but they change charges. So they'll change a glutamic acid to a lysine in the triple helical domain and give rise to a vascular EDS-like, but not real, not nearly as much involved in vessel disease, but they have skin abnormalities and intramuscular bleeds and things of that sort. So it's worth asking again, and it's worth asking a laboratory that does stuff. And so you can certainly write to us at the Collagen Diagnostic Lab and ask what's going on. And we handle that a lot.
Okay.
So VUSs often become benign or often become, and less often become pathogenic. They become pathogenic usually for something that's a little bit different in terms of the phenotype.
Very helpful. Thank you. What do we know about how a pathogenic mutation in FBN1 affects the severity of the disease?
Well, it's interesting. So the null mutations in COL3A1 are milder. That is, they have a longer a dormant period before things begin to happen. The life expectancy is longer than people who have most missense mutations and most splicing mutations. It's been a little bit more difficult, and I had thought that the same would be true for fibrillin, but it's not. And it looks like null mutations in fibrillin, heterozygous nulls, are more severe, have earlier onset than others. Substitutions of cysteine in the 6 cysteine domains tend also to have earlier onset than others. And so mutations outside of those are less severe. So that sort of thing is being cataloged now, and more recently there's an attempt to really try to define that. The cysteine substitutions were recognized early on by Uta Franke at Stanford, and it became clear that they were more likely to be associated with things like lens dislocation. But we've found that mutations along the gene can be quite different, and in some people they have only skeletal changes, sometimes they have only ocular changes, and sometimes they have only cardiovascular changes without any of the associated things that we see otherwise. So mutations in the 8 cysteine domains, or the TGF binding like domains, tend to be milder than the ones that are in the 6 cysteine domains or calcium binding EGF domains. We don't know much about the difference between mutations in the calcium binding domain as opposed to the other portions of those regions of the molecule. But it's pretty clear that there's a fair amount of heterogeneity. There's a lot of heterogeneity in the mutations, and there is probably a fair amount of heterogeneity in the clinical response that I think we didn't appreciate to begin with. Didn't appreciate it to begin with because early on we just sequenced people who had relatively severe phenotypes.
Makes sense. Are all genetic testing labs created equal when it comes to panels for a genetic aortic condition? So should folks expect that Invitae is running the same protocol as CTGT and so on?
I think almost everybody is now using pretty much the same kind of technology and doing things the same way. They don't necessarily, so for exomes, you have to capture the sequences, and they may use capture probes coming from different places. They may be able to read whether there are, pieces missing or not more easily than others. But fundamentally, they're doing the same test and they're doing the same kind of thing. The technology has been standardized now to doing the same thing. And the next generation of testing will move from doing short read sequencing. The sequences that are being sequenced now are about 125 to 150 nucleotides long. And there are now ways to read the whole gene in one fell swoop using long read sequencing. And that's being used. It's much more rapid. The preparation for DNA is much easier, and the turnaround times can be very short. So it's possible to sequence an entire genome if you have the right equipment and the right stuff to do it in a few hours as opposed to days. I mean, it's a huge difference. From 13 years to a genome to 6 hours to a genome is several big strides.
Yeah.
And getting it into the clinic is a little more difficult, but it's being worked on.
This person indicates that their aunt and their brother both had genetic testing done. Both have the FBN1 gene of the same pathogenic variant, same protein, everything the same. And this person's asking if they have to be tested as well. Their doctor is pushing for it, but they've had a diagnosis since they were 4. They don't want redundant tests if it's not going to change their treatment plan.
Well, it's an option. It depends on. So, what do you want the testing for? Is people with the same mutation will differ in their clinical pictures because the other 20,000 genes can also modify what the phenotype is. if you're interested in having children and you want to determine whether you can select select embryos that do not have the one, then genetic testing is necessary to show that you do in fact have the same. I mean, the likelihood that you have the same mutation is very high, but just to be sure, no genetic testing company that's going to do pre-implantation genetic testing is going to do it without you being tested. So that's probably the major issue, I think, in doing testing.
When do you recommend someone having updated genetic testing performed?
Well, if you've had something identified, you're home free, I think. If you have if you have a clinical phenotype that is like Marfan syndrome or like vascular EDS or just aortic diseases, and something hasn't been done, hasn't been found up to this point, then the number of genes that are being looked at is changing every year. The technology is changing, so that it's much more likely that we would do either whole exome or whole genome sequencing at this point than to do panel testing. And so, that probably within 4 or 5 years, it's reasonable to ask for being reconsidered if you're trying to find something that hasn't been found. If you have something that's been found, you probably don't need more unless you're asking a different question, which is there a difference? Why is there such a big difference in presentation between me and my brother and my father? And there you're really asking a research question. What are the modifiers? What are the influencers that are acting there at that point? And that really is something that would be done only in a laboratory where people are sort of studying things.
Okay. One more we
Mhm.
have time for. Someone noted your generous offer to field emails
Yeah,
by sharing your email address on the screen. Does that apply to folks outside of the US? This person specifically from the UK.
yeah, we've done that. So in the UK, there are excellent laboratories at Sheffield and London that work on these conditions, and I would try that. I don't know how responsive they are. We've always been responsive to people asking. My email address is incomplete. It doesn't have a U at the end of it, I noticed. So it's pbyers@uw.edu. But the best thing to do is actually to write to the Collagen Diagnostic Laboratory and get the email address for there. And it would go to either Drew Leistritz or Ulrika Schwartz, who's really taken over at this point. But we will continue to share them and look.
Thank you.
You're
So
very
many
welcome.
thanks to those who shared their questions and obviously to Dr. Byers for so thoughtfully addressing them. If we didn't get to your question, please know that staff in the Marfan Foundation's Help and Resource Center can assist you with questions about your specific situation. Please direct your questions to marfan.org/ask and someone will get back to you.
Right. And if you summarize those, we can work together to try to answer them
Wonderful.
as well.
Okay, let me just share my screen one more time as we wrap up. So our webinar tonight is the second in a 3-part series on genetic testing. Please join us again on March 21st for a discussion with Dr. Bart Loeys on variants of uncertain significance. You can register for this webinar by scanning the QR code on your screen. We'll also communicate information about the webinar by email and on our social media channels, so we hope to see you next month. And one more thing. Please take a moment to share your feedback on this webinar by visiting marfan.org/survey or scanning the QR code on your screen. If you don't get a chance to scan the QR code tonight, you will receive a survey by email when the webinar recording is shared in a few days, so be on the lookout for that. Thank you so much to Dr. Byers. Thank you so much to all of you for being here. Take care.
And thank you all.
Thanks, Peter. Have a good night, everybody.
Yeah.